November 2011. In the University of Toronto's Department of Biochemistry, a researcher is about to make an astonishing discovery. Two pipette tips had fused, forming an aberrant oligomer. Although innocent-looking, this oligomer possessed a strange property. When added to a container of similar particles, much to the researcher's surprise, a spontaneous polymerization reaction occurred. In the coming weeks, the researchers would examine the strange pipette-tip fibrils carefully. Their unusual size presented a unique challenge, however. They proceeded to examine them by microscopy, spectroscopy, telescopy, and scopy. They found that the fibrils were resistant to boiling, heating with detergents, treatment with proteases, and ultraviolet irradiation. Most importantly, they discovered that any part of a preformed fibril could be used to seed the polymerization of a fresh batch of pipette tips. This led the team to hypothesize that the large-scale phenomenon that they were observing was related to the microscale prion phenomenon, in which a protein converts to an aberrant conformational state. In this state, it can recruit benign protein molecules to associate with it and form a fibril, converting them to the aberrant state in the process and conferring on them the ability to convert further protein molecules. Consistent with this, the team found that the large-scale fibrils stained intensely with the dye Congo red. Under crossed polarizers, the stained fibrils showed the same green birefringence characteristic of the amyloid fibrils formed by the prion protein. Although no one had ever seen the prion phenomenon occurring on this scale before, the researchers soon realized that many examples of macroprions, as they titled the phenomenon, existed in nature. Theoreticians and experimentalists collaborated closely to try to model the assembly mechanism for the fibrils. They were able to rule out parallel, anti-parallel, and tip-to-tip models, eventually discovering that a tip-to-tail model was most consistent with experimental findings. The findings were well received. However, one troubling question still remained. The ability of microscale prions to convert benign particles provides a new mechanism for infectivity, and this was the basis for the mad cow disease outbreak of the 1980s and 90s. Could macroprions present a similar danger? The team found that, like their smaller counterparts, macroprions could trigger self-assembly of dissimilar particles, albeit with very long lag phase kinetics. Proceeding to animal trials, they found that hamsters were highly susceptible to macroprion toxicity. Similar results were reported for mice, koalas, and baby seals. Other common laboratory organisms, such as giraffes, were largely unaffected, but for some frothing. At this point, the lab lost its ethics committee approval to work with animals. Consequently, the team next turned to human trials. Humans proved resistant to the effects of macroprions.
Stop that!
Despite this, laboratory contamination soon became a problem, hindering productivity.
Oh shit!
Before long, the situation became desperate. Drastic decontamination action was needed. Fortunately, thanks to the quick and selfless action of these brave researchers, major disaster was averted. For if even one of these macroprions had escaped into the environment, who knows how much chaos it may have caused. Thankfully, that frightening scenario will never come to pass, and the macroprion phenomenon will remain nothing more than an interesting curiosity.
Guys, I don't think we're getting the deposit back on the giraffe.